How's it going, you guys? So in this video I wanted to discuss mutations. So mutations are any change in the genetic material. They could lead to good changes, they could lead to bad changes, depending on how they affect the organism. But really, mutation is any change in your DNA, in your genetic material. There are a couple different types of mutations. There are point mutations, which are changes in single nucleotides. So let's say you have a sequence of like AACTG. Maybe we change it to AAGCT or whatever I said. So basically, a point mutation is a change in a single nucleotide, right? Whereas chromosomal mutations are larger changes. They are changes in the number of chromosomes. So if you get too many chromosomes or inherit too many chromosomes, that could be a chromosomal mutation. But also the general structure of the chromosome. So let's say maybe one chromosome ends up getting a whole nother chunk of chromosome. So chromosomal mutations come up during meiosis. If you remember crossing over leads to chromosomal mutations. If they don't separate properly during meiosis, that could lead to chromosomal mutations where there are too many or too few chromosomes, leading to something like Down syndrome. But point mutations are errors in DNA replication. So when we split the DNA apart and we're copying both strands, maybe we see an A and we're supposed to put down a T, but instead of putting down a T, maybe we accidentally put down a C. And that's done by DNA polymerase, if you remember that enzyme. So point mutations are changes in single bases, single nucleotides, while chromosomal mutations are errors that arise during meiosis. So I don't want to focus too much on chromosomal mutations, but I want to focus mainly on point mutations. So point mutations, changes in single bases, single nucleotides, they have a couple of different ways, or we have a couple different ways that we could do this. We could have substitution mutations. That's where you're supposed to put down one base, one nucleotide, but instead you put down another. So you're basically swapping one nucleotide out for another. But we also have frameshift mutations. Frameshift mutations are insertions or deletions. So maybe we accidentally put down one too many nucleotides, so we insert one without taking one out. Or deletions, we remove a nucleotide, don't put one down, and don't add another one in its place. So substitutions, you swap one out for another. Frameshift, you either insert one or delete one without replacing it. So let me just explain this a little bit more with pictures. So here we have our sequences. So let me put it like that. Sorry for the glare right here. But here, this is our DNA sequence. So I'll put DNA. This right here is our mRNA. And this right here is our protein, our amino acids. So if you remember, we have transcription and then translation. Transcribe it into mRNA, then translate it into protein. So with this in mind, the original DNA sequence said TAC, GCA, TGG, AAA, which means that we're going to get this mRNA sequence where T pairs with A pairs with U because it's RNA, C with G, etc. So we have DNA to mRNA, and if we read these mRNAs in groups of threes that we call codons, if you remember, we have codon AUG, codon CGU, ACC, UUU, those are our codons. They translate to one amino acid. So AUG translates to MET. That's our start codon, so we translate to our MET, or methionine. So that's our protein. Now, I want you to imagine a couple scenarios. So for substitution, maybe right here where we see GCA, instead of putting down a C, we actually put down a T. So we swapped out C for T. So that is a substitution. So that means that we're no longer going to get C to G, but we're rather going to get T to A, and if we have CAU, that's a different codon than CGU, right? So CAU translates to his, whereas CGU translates to ARG. So here we see a single amino acid changed in the final protein. So hopefully everyone sees that. So a substitution typically leads to a single amino acid change. Sometimes it could lead to no change at all, which we call a silent mutation. That would be if we swapped one nucleotide out for another, that led to a different codon, but that codon, sometimes multiple codons, translate to one amino acid. So we could have multiple codons, maybe we changed it to a codon that still translates to the same amino acid, so it's a silent mutation. But oftentimes it just swaps one amino acid out in the final protein. So hopefully everyone sees that. Now compare that to insertion and deletion, which again are both frameshift mutations. Frameshift. We call it that because they shift the entire frame that we're reading, the reading frame. So we read it in groups of threes. So if we were to insert a single nucleotide right here, now we have completely different groups of threes. So let me give you an example. Right here, TAC, we have TAC, but right before GCA, we insert an A. So that's an insertion. So now we have a U in the mRNA where we didn't have it before. Notice we didn't remove anything when we inserted a new base, a new nucleotide. We didn't remove anything, so now that changes what we read. We are no longer reading CGU, but we're rather reading UCG. We are no longer reading ACC, but we're now reading UAC. So everything shifted over one, which means we have all of these new amino acids. So insertion leads to one nucleotide, all the nucleotides shift down one, and now we have completely new amino acids. So insertion, deletion does the same thing, but just instead of shifting forward, we shift back. So here, imagine we have GCA, but we removed the G. So we took out the G. So now we have CAT, which translates to GUA. So everything shifted back one. So now we have different codons that we're reading. So instead of ARG, we now have VAL or valine. Instead of thur or threonine, we now have pro or proline. So if you compare the two, if you compare substitution mutations, where we swap one nucleotide out for another, and compare that to frameshift mutations, you can see that frameshift mutations tend to lead to more severe outcomes, more severe mutations. So frameshift mutations change everything down the road, and that will ultimately lead to a greater change, typically, in the amino acid sequence. So hopefully we see the differences here between the different types of mutations. I'll take a step back just so if you wanted to pause this and look at these a little bit closer, you can. But I'm going to talk about the last thing about mutations, which is good versus bad mutations versus non-affecting mutations. So some mutations that arise are good mutations. Typically, that's not the case. Typically, most mutations either don't affect you at all or could be bad mutations and lead to genetic diseases, such as like cystic fibrosis, if you've ever heard of that. But an example of a good mutation, so however long ago some humans migrated north, right? So starting off in Africa, we gradually migrated north, and we had humans in European areas. And as you go north, there's less and less sun, right? So the sun dies down, we get less of it. So really it's not as beneficial. It's beneficial in Africa to have dark skin because it protects your cells from UV radiation damage. But when you go further north, it's actually not as beneficial because the darker skin prevents you from absorbing what little sunlight there is, what little UV light there is, and we use UV light to make vitamin D, which helps our bones absorb calcium, right, and get stronger bones. If you ever heard your parents tell you to go outside because you need to get some vitamin D, that's because you go outside, you get the UV rays. UV rays helps your body make vitamin D. So further north, it's less of an issue of skin cancer and DNA damage, but it's more of an issue of not getting enough vitamin D. So there was a mutation that came up which actually caused the skin to get lighter, to not make as much melanin, which is the pigment that makes your skin darker. So once you got that mutation that actually caused you to not make as much melanin, not make as much pigment in your skin, those people were better off. Those individuals were better off because they were in more northern countries, which tend to get less sunlight. And so they were able to absorb more of that sunlight and get the vitamin D they needed. So that would be an example of a good mutation. And that's a good mutation relative to where they lived. A mutation that causes you to lose skin pigmentation would be bad in Africa, right? So that same mutation could be good one place, but bad somewhere else. But an example of a bad mutation, regardless of where you are, is a genetic disease mutation, such as sickle cell anemia. That's where your cells actually become shaped, your red blood cells become shaped like a sickle. They cannot carry oxygen as well, which means your body can't be pushed. You can't really push yourself, work out too hard, because sickle cell anemia, if you have that, your blood cells have a hard time carrying oxygen to other parts of your body. So that would be an example of a bad mutation. But most mutations, I would say, are non-affecting, so they don't really change the individual, at least change them enough to make them, I guess, not function as well or function better in their environment. So overall, just keep that in mind. Mutations could be good, bad, or non-affecting, and that's pretty much it for mutations. So if you have any questions, let me know, leave a comment below, or send me an email, and that's pretty much it.